Dissolution test specifications, basic understanding, and why viewpoint limit is like 70%, 75%, 80% or 85%. This is the main topic or main motto of this video. Welcome dear viewers to this interesting video. It will be a short video to understand the basics for dissolution test specifications. Why Q point limit is not like 100%? Because as we give the label claim and we denote it by testing assay, that is 100%, and the limits for assay or potency are 95 to 105% or 90 to 110% or 110%. But if you consider the dissolution Q point limits, these are generally given as 70%, 75%, 80% or 85%, and Q is denoted as Q is equal to not less than 80% or 75%, 70% or 85%. Let's start with the video to understand these terminologies. Dissolution test specifications and why Q point is 70, 75, 80 or 85%. See, mainly for dissolution testing, dissolution specification is very important, as this limit will decide whether the formulation is passing the criteria or not. And achieving the optimal dissolution specification in the pharmaceutical product is prime importance. Exploring practical considerations for effective drug formulations. This will be the objective. The goal of dissolution in the pharmaceuticals. So what is the goal? Why we do the dissolution testing? 100% dissolution often seen as the ideal, but practical considerations make 75 to 80% dissolution more feasible. As I have mentioned, 100% label claim should get dissolved, but generally 70 to 80% is the feasible one. Dissolution is essential for bioavailability, regulatory compliance, product performance, and quality control. Generally, the dissolution should be 100%, as it will denote that the complete drug is dissolved and is available for the systemic action. But there are certain challenges, there are some problems because of which we cannot achieve 100% dissolution for all the formulations. It may be possible for some of the formulations, but generally it is not feasible. And why this happens? Because of the manufacturing variability, then particle size and particle size distribution of the APIs, and the solubility limitations. Manufacturing variability, tight control required for complete dissolution. It will be a costly and it will be difficult. Particle size, that is PSD and distribution. Certain particles may not dissolve fully under optimal conditions. Generally, the dissolution conditions should be mild, should not be much aggressive. So you are achieving the 100%. And 100% dissolution is not the goal. Then solubility limitations are there. Active ingredients may have inherent solubility restrictions. Some APIs may float into the bowl. Some may get little bit degraded. Sometimes the APIs may show solubility problems in the media, and that's why always 100% dissolution will not be possible or feasible. Bioavailability and regulatory considerations. So for bioavailability, 75 to 80% dissolution often ensures sufficient absorption and efficacy. Regulatory guidelines are there, as FDA recommends dissolution levels of 75 to 80%, aligning with bioavailability needs. So that's why 100% dissolution is not required, or it is not feasible, and it is not suggested by the regulatory bodies. Then you consider the cost benefit analysis. Dissolution run for IR tablets is generally for 1 hour or 1.5 hours, sometimes it is 2 hours. And if it is not showing 100% release in 2 hours, for example, then it will go for a longer time. And it is not possible that if you run the dissolution testing for a longer time, it will give you 100% release. So it will add cost and it will not have any benefits. Process reliability and flexibility. Process reliability, allowing a margin in dissolution specs, ensures smoother manufacturing and fewer failures. If you make it very tight, that time number of batches may get failed, which are otherwise passing in vivo. Regulatory compliance, slightly lower resolution specification, align with the manufacturing efficiencies and regulations. Role of dissolution methods. So, what is the main role? First is method strength, second is purpose of dissolution and other tests. Method strength means dissolution method should not be too aggressive to lose discriminatory power. If you are trying to achieve 100% dissolution and you are making the dissolution parameters unrealistic or bio irrelevant, then it will become difficult. So always dissolution testing should be performed with milder conditions or standard dissolution conditions. I have made many videos on these topics, so you can go through the playlist and watch those videos. So here just I have mentioned that dissolution method should not be too aggressive, or uh, the parameter should not be such that it lose discriminatory power. Purpose of dissolution. Dissolution testing is meant to show the performance of the product and not just its contents, because testing for the content and content uniformity is in the assay test and content uniformity testing. So other tests like assay and content uniformity helps to assess API extraction and quality, but here 100% is not the requirement for dissolution specification. BCS class you can consider here. BCS class 1 and 3 are highly soluble molecules. BCS class 2 and 4 are low soluble. So regulatory bodies expects rapid and very rapid dissolution for effective bioavailability. Complete dissolution is more critical in these classes. BCS class 2 and 4, low solubility, low permeability. So complete dissolution might not be feasible and 100% dissolution is not always necessary. Now coming to the IVIVC. IVIVC can give you the information regarding the in vivo behavior and in vitro in vivo correlation. IVIVC can justify lower dissolution specifications if in vitro performance correlate with adequate in vivo bioavailability. It is not like that if you are getting less dissolution of 70% in vitro and you will get the same in vivo. In vivo it may be improved for some classes. Plus BCS class 2 and 4, lower dissolution, example 70 to 80%, can still be therapeutically effective if linked to the in vivo data. And if you try to go to get 100%, it will not be feasible because of some problems, some challenges, as I have mentioned in the earlier part of this video, like solubility challenges, manufacturing challenges, and the particle size and particle size distribution challenges. Now see the regulatory flexibility. So FDA guidance is there, ICH guideline is there. These guidelines specifies varying dissolution targets based on the BCS class. So for class 1 and 3, these are highly soluble molecules, require higher dissolution like 85% or more than that. However, class 2 and 4 allow more flexibility based on solubility permeability profiles. Global variations. Different agencies may have stricter or more flexible requirements. Now relate these things to the discriminatory power of the method also. Dissolution method should maintain discriminatory power to assess performance. So we should not try for 100% dissolution in all the products and at all the conditions. Avoid overly strong methods. Too strong method may not reflect true bioavailability or therapeutic outcomes, especially for poorly soluble drugs. So it is not like that. You are not getting 100% dissolution means it will not be effective in vivo. It is not like that, as we have seen this in IVIVC part. So in conclusion, the dissolution specifications are very important and these decide the discriminatory power, IVIVC, bioavailability and the product quality and the product performance. So setting the right dissolution specifications is very important and its basic understanding is required. It is a balance between bioavailability, manufacturing reliability and regulatory requirements. Flexibility in dissolution targets allows for optimal therapeutic outcomes without unnecessary cost and challenges. So once you get the 70%, 75%, 80% or 85% dissolution, you can give the Q point. The Q point may be for IR products like 15 minutes, 30 minutes, 45 minutes or 60 minutes. Sometimes for some of the products, low soluble products, 90 minutes, 120-minute Q point may also be given. Sometimes two Q points are given for the low soluble molecules like 50% dissolution and 80% dissolution as two Q points. So I hope this video will help you to understand dissolution test specification basic understanding and why 100% dissolution is not required or recommended or feasible, and why you can give the Q point limit of 70%, 75%, 80% or 85%. Thank you for watching the video and let's meet soon with such kind of informative video. Thank you.